The overall goal of this procedure is to assemble a continuous culturing vessel in which the response to illumination of optogenetic microbes can be measured with a microscope automatically in real time and over multiple days.
This method can help answer key questions in cellular biology and metabolic engineering, such as how dynamic gene expression can elicit responses which differ from static gene expression.
The main advantage of this method is that continuous culture enables measurements to be collected for multiple days.
To begin, solder the ground line, the data line, and the positive voltage line of the digital thermometer to the printed circuit board. Clip off one pin from a female 3-pin header and trim the remaining 2 pins. Solder this in the pair of holes labeled R2 and connect the 2 soldered pins by inserting a 4.7 kilo-ohm resistor in the pin header. Next, connect the remaining components to the circuit board, overlay the circuit board onto the microcontroller, set up the light-proof enclosure, and install all software as described in the accompanying text protocol. First, place the long end of the aluminum port through the gasket into the culturing vessel and screw on the cap. Cover one short aluminum port outlet with medium diameter silicon tubing. Plug the distal end by inserting a female lure lock and then connecting a male lure lock plug. This outlet is supplementary and will not be used. Connect 2 short segments of medium diameter silicon tubing with male and female lure locks. Then, connect one end to a short